I will be briefly talking about the research updates and also diseases, what we are seeing in our research orchard here in Geneva. First, I would like to show you this slide where I'm showing you data from apple scab in 2018 and 2019. And this is an unsprayed apple orchard where we have about 200 different accessions of wild species as well as cultivated apples. And this data was taken in the orchard where we don't spray, we don't manage anything. There's no fungicide, there's no antibiotic spray. So basically the idea was to see natural resistance against apple scab in these 200 accessions from cultivated and wild species. And the scale we used was 0, no infection, 1, weak infection, 2, as moderate infection, and 3, highly susceptible. And I'm showing you here just a snapshot of a few cultivars. And what you can see here in this graph that most or many of the cultivars like Macintosh, Granny Smith, Room Beauty, Cortland, they have high level of scab infection in 2018 and 2019. And as you know that there was a lot of infection in 2019. So that is also obvious in this graph, the green bar and green, blue, and orange bar. But what I want to highlight here that although those susceptible cultivars were showing a lot of infection in 2018, all those 3 time points, and also 2019 in 3 time points. But there are some cultivars you see here, for example, Golden Delicious, Jonathan, that they were holding up against apple scab, even with this high inoculum pressure. But when in 2019 there was a lot of inoculum, they were not able to resist and they fall into highly susceptible category 3. But if you look on far right side, and you can see Florina, Caesar Jersey, Prima, Jonafree, Redfree, some of those varieties, and they didn't show any infection, although there was so heavy in inoculum pressure in both 2018 and 19. So what it actually points to suggests that there are scab-resistant genes which are present in wild species or also in cultivated sources, noun-resistant genes, which can be bred to develop resistant cultivars. So I think many of you already know that, but I want to emphasize on that again. And also, I think there are few wild accessions and few cultivated, domesticated accessions we found. They were not reported previously that they carry a resistant gene. So we need to really go ahead and identify the genes behind that resistance so that breeding program can use it for breeding resistant cultivars. So here in this next slide, I'm showing you the pictures of disease symptoms or observations from 2020 in May. And one big picture here, you can see that there is everything here in upstate New York and in New York State and in Geneva. So first scab infection we observed in this orchard in early cultivar called Anna from Israel on 17th of May. And then also we see a lot of infection now on Gala and many other cultivars. Similarly, we see now as Art was mentioning about petal fall. I mean, at this stage, you can see that there is a lot of primary infection, and it is maybe too late to really do anything at this point. But I think what it suggests that one has to be proactive, and depending on the maturity of these varieties and cultivars, one should do different managements maybe. Also, you see on Jonafree, either Jonathan, a lot of powdery mildew. Another thing I want to mention here is the frog eyelid spot. So as I said, we didn't manage this orchard, but if there were mummified fruits or dead branches or dead trees, we see um, a lot of frog eye leaf spots or this black spots you see in the middle. And they're already, it's too late. So I think sanitation is very important. And if there is any leftover, dead wood or dead tree or anything left from pruning or from fruit thinning, I think it's really important to remove that. And also on extreme right side, what you see, there are some cultivars, although there's so much inoculum for powdery mildew, scab, frog eye leaf spot, and many other things, they were still clean. And especially one thing I want to point that the susceptible cultivars are the cultivars which the side of the tree which was closer towards the dead tree showed infection, but on the other side it was pretty clean. For example, in the bottom right picture is Empire. Because Janet told me to stick with 3 slides, so this is the last slide. And here I want to give you update what we've been doing on research side. So on research side, as you know, my program is looking for genetic basis of resistance and to find genomic regions in the genome of apples which are linked with disease resistance, especially fire blight and apple scab, so that breeding program can use those DNA markers for breeding resistant cultivars. So we have genetic mapping populations, and we have been inoculating these in May, in this month, in the greenhouse with fire blight. So we have pedigree or mapping population crossed between 2 parents, and these are 150 siblings in 4 applications, and we see very nice inoculation there and very nice segregation. Some are very resistant and some are very susceptible, and that's what we need to do genetic mapping to find the genes behind this resistance. In the bottom graph, what you see, we are also running a computer vision competition at Google's competition platform called Kaggle, to classify foliar diseases of apples. And this was for first time we ran this competition. It was based on disease images, especially foliar diseases from last year. And we have created a pilot data set with about 4,000 images of 4 foliar diseases and healthy leaves. And we made this data available, or these images available, to global community of computer scientists at Google, so that they can compete and come up with the best prediction model to classify foliar diseases of apples. So this is for the first time, but it will take a couple of years, iterations, and we have to make a stronger data base, so that this can be used for automatically classifying diseases to help scouting of diseases. So at this point, there were more than 1,300 teams competing to make the best prediction model, and they submitted almost 6,000 prediction models, and the best model so far is predicting with the accuracy of more than 99%. So very high accuracy, but we need to really strengthen and make the bigger database. And I need also support from you, if you can take some images while you're walking around, and it can help to strengthen the database. So with that, I would like to thank you.